Everyone for joining us for our first monthly seminar of 2026. My name is Benjamin Keene. I'm here at the Center for Physical Genomics and Engineering at Northwestern University. We host this seminar every month. We're really excited to kick off the year with our guest speaker, Professor Taekjip Ha. Dr. Ha is the George D. Yancopoulos Professor of Pediatrics at Harvard Medical School and Director and Senior Investigator of the program in Cellular and Molecular Medicine at Boston Children's Hospital. He's been an investigator with the Howard Hughes Medical Institute since 2005. He received his PhD in physics from the University of California at Berkeley. After postdoctoral training at Stanford, he was a physics professor at University of Illinois at Urbana-Champaign, where he co-directed an NSF Physics Frontier Center, and he was the Bloomberg Distinguished Professor at Johns Hopkins University. He's a member of the National Academy of Sciences, the National Academy of Medicine, and a fellow of the American Academy of Arts and Sciences. I could go on listing his accomplishments and awards, but we wouldn't have time for a talk if I did that, so I will just say that we're very honored to have you, Dr. Ha, and thank you so much for taking the time out of your busy schedule to share your work with us. Just a quick note for our audience. We will have Q&A at the conclusion of Professor Ha's talk. So if you have questions, feel free to type them into the Zoom chat at any time. Or at the end, if you just want to raise your hand, turn on your mic, we'll do questions then. If you want to learn more about our research and training here at CPGE, you can visit us at physicalgenomics.northwestern.edu. Or we have a YouTube archive of all of our talks and seminars. You can follow us on Twitter X or Blue Sky. So please do follow up and check us out. At this point, Dr. Ha, I will turn the Zoom over to you. And thank you so much again for doing this for us.
Yeah, thank you. Thank you for having me and also for organizing these important community building activities offline and online. It's my great honor to talk about some of the work that we've been doing in the lab, some unpublished, some published. Let me share my slide here. I actually wrote this title for this talk as DNA and nucleosome properties as a biophysical backdrop for genome function, that these are intrinsic properties of these molecules as they are physical entities, not just a string of letters. And whatever nature comes up with will have to contend with their biophysical properties, and that's the idea here. A few months ago, I decided to think of the title for my laboratory's research, and this is what I came up with, and this is what AI produced. Genome maintenance at high resolutions. We are interested in studying how genome is duplicated accurately and maintained through DNA repair when there's a damage. And we are trying to do this at higher resolution in time and space, single cell, single molecule resolution, and perhaps most importantly, higher the resolution than yesterday. So our genome is organized through multiple layers. We have nucleosomes with 147 base pairs wrapped around the histone core, and some of them may form clutches of few nucleosomes. And we have loops between enants and promoters making contacts for controlling gene expression. And we have a larger structure such as tags and compartments that I will discuss later today. But before we begin, let me just remind you that information is encoded in DNA and also in the nucleosomes. For example, DNA sequence encodes at primarily amino acids in the coding region. And you can also change the gene activities by changing transcription factor binding sequences in the genome. In terms of epigenomes, DNA methylation, histone modifications, they can act primarily working with transfactors, readers and writers and rearrangers to control the genome organization and genome function. But today I want to talk about perhaps additional layers of information contained in the DNA and the epigenome in terms of DNA bendability and nucleosome condensability that can form the biophysical backdrop of genome function. So one tool that we love is optical tweezers. I call this chopsticks are made of light. You can use a focused laser beam to grab a single particle. And if you apply force using the laser, then you can play a tug of war with a protein sitting on a glass surface through a DNA tether and measure these activities very precisely down to a piconewton of force. And actually, this is not a new idea that you can extract information by stretching. In fact, in human history, we have learned that you can extract information from, stretching involuntarily by asking questions such as how flexible is DNA. So in my lab, we also are known for a technology called a single molecule thread, and where you can put dyes on the DNA, green and red, and measuring their relative intensities and infer relative distances between the two dyes. When you have two dyes close to each other, when you excite the green one, there is a strong energy transfer to the red, strong red, and then you get a red signal instead of green signal, and so on. So many years ago, Sang-Chul Hong in the lab combined optical tweezers and single molecule thread. So you can measure conformational changes using thread, but as a function of applied force. And a student in the lab, Robo, who is now in the faculty of Penn State Chemistry, used that instrument to measure DNA unraveling from a protein surface. In this case, a single-strand DNA wrapped around a protein called SSB. And we label the DNA with the dye pair so that at low force, you get high thread from the fully wrapped structure. But as you increase the force, distance should increase, and then you get less and less thread, and you get mainly a green signal. In fact, when Robo use optical tweezers to apply increasing force and coming back and do it 5 times, you can see the red signal is going down and green is going up gradually due to a gradual decrease in thread coming from gradual unwrapping of the DNA from the protein surface. And you can also show that thread efficiency also goes down gradually. In fact, when he showed me the data, it was so beautiful, I almost cried. Although I didn't actually cry, I told him to go back to the lab and take more data so he could actually publish as a nice paper. And then this actually is ask us, basically we ask, well, maybe you can also do the same thing using the nucleosome because nucleosome is just a DNA wrapped around the protein core. But if you look at the nucleosome, there's a really sharp bending of DNA. And in fact, highly bent DNA is found in many other places in life. And we thought maybe this is something that is sequence dependent. And if the sequence makes DNA more bendable or flexible or cyclizable, then maybe you can form the circles more readily and nucleosomes may form more easily. So in that sense, you can think about an idea of a mechanical code where if you make a mutations, of course, you'll change the genetic code and you'll change the transcription factor binding to change the cis regulatory elements, but you may also change DNA mechanics and that may act directly, at least in certain non-coding DNA context. Many years ago, former student Reza, he is now on the faculty at Northwestern, developed this single molecule looping assay or cyclization where 100 base pair of DNA can be induced to form a circle through base pairing between the two sticky ends. And this is as you add high salt. And you can use thread to measure the kinetics of looping. And we deduce that faster looping is caused by more bendable or flexible DNA properties. And later on, another student, Tui Nyo, she used these optical tweezers to apply force to unwrap the DNA from the histone core. We measured it using Fred, and she found that DNA unwrapping from the core was actually not symmetric. Yeah, one end unwrapped more easily than the other at a lower force. And she was able to use Raja single molecule looping assay to make the connection so that the less bendable DNA segment, according to Raja's assay, unwraps more readily, likely because more bendable sequence actually stay bound to the bent form to the histone core at even higher forces. So these are really nice studies that we examined many years ago. And the questions that arose are the following. Sequence features are actually important for DNA bendability. Then what are the rules? And if bendability is important for function, then in the present-day genome, we should have also signatures that arose throughout the millions and billions of years of evolution. So for that, former postdoc, Akash Basu, he developed this loop-seq method. If you want DNA physics meets next-gen sequencing, here we have a library of many different sequences. I'm showing you only for 4 sequences, and each sequence has many copies. And then you add Searle to induce looping, and DNA sequence 1 that is the most flexible, more bendable or cyclizable will form circles readily, whereas the most recent sequence stays linear. And then you can use an exonuclease that will digest only the DNA with exposed ends. And then you can enrich for sequences that have high bendability or flexibility. And then by doing next-gen sequencing and comparing to the input, we can quantify the survival probability. And we simply use a natural log of that as a measure of flexibility or bendability of each sequence in the library. And we can do this from up to 90,000 different sequences at a time, or 50 base pairs in length. And in the paper, we included lots of sequences. And one interesting finding that we included in the paper was the following. So in gene promoter regions, we have regions that are depleted of nucleosomes. And because the histone has DNA sharply bent around it, perhaps if DNA is more rigid due to sequence properties, maybe it's easier to form these NDRs. And in fact, if you measure this cyclizability, that this is going to be the name that I'm going to be using today, cyclizability across these promoters, average of more than 500 genes, and we see a nice dip in the promoter region, exactly coinciding with NDR, where nucleosome occupancy is minimal, suggesting that perhaps there is a mechanical component in determining and defining this critical region of nucleosome depletion. Then we worked with a student in Korea, Jong-han. He developed this machine learning algorithm to train convolutional neural network using a subset of data. And then using that, because we have the sequence and cyclizability, the C mapping, we can then use the algorithm to actually predict and compare to the measured, and using the withheld data set, and we can get better than 95% accuracy. It's so good that we no longer have to measure this anymore. And then you can actually begin to do some interesting exercises, and I'm going to show you just one example on the right side, engineering DNA mechanics. So what he did was to start with a random DNA sequence of 50 base pairs in length, and then he created all possible single point mutations. So it'll be 50 times 3 different mutations per site, so 150 sequences. And for each sequence, you predict a cyclizability, and then you pick the winner sequence with the highest value of cyclizability or C value, and then you provide that as an input for the next round, and you do this many times. And in fact, if you do that, you can quickly shift the distribution of the cyclicity center around zero for random sequences to something uh, very extreme in terms of bendability. And you can also do minimizing selection and go to very rigid sequences. You can see interesting patterns that are kind of periodic. Although for overbendable sequences, we evolved, GC content is actually about 50%. It's not really any special for these molecules. And because these values are so extreme, they're not found in our training set. These are these chromosome sequences. So we wanted to validate this by doing some single molecule measurements. So Jingzhou in the lab, she performed single molecule cyclization based experiment and showed that indeed this very flexible sequence forms a circle within a minute, whereas the most rigid sequence rarely forms a circle even after an hour, validating that these methods actually working. And Ting Wei then also used AFM to provide additional validation for highly cyclizable DNA versus rigid DNA, and some quantifications are shown here. One more thing that Jonghan did was to pick the champion top sequence, and he actually did something crazy. He put it into the database and ran blast nucleotide search to see whether there's anything found in nature. And he found matches, and these matches are shown here, and all from a single organism, Neisseria karyoxanus. And this is a single cell eukaryotic organism living in the sea water. And the sequence is found many, many times in the mitochondrial DNA, which is a non-coding region of this organism. So that was very surprising. We thought we just created something very artificial, but nature actually got there first. Older, but this is collaboration with Julius Lux in Czech Republic. And interestingly, this organism has more DNA in mitochondrion than in the nucleus. And in comparison, human cells have actually 1,000 times more DNA in the nucleus than in mitochondria. So this is very unusual. And also, this organism has lost mitochondrial DNA compaction protein, known as proteins that contain HMG boxes. So one idea is that there's a evolutionary pressure to pack so much DNA without the help of a DNA compaction protein may have led to the highly bendable sequences basically occupying much of the genomic mitochondrial DNA space. And in fact, this highly bendable sequence that we discovered from computer simulations and then found here, and this is a consensus sequence found in this organism, and there's some natural variations. But if you look at the natural variations and predict their cyclizability C value, they are still all very high between, this value of 2 and 3. That's a pretty large number because it's a log value. But if you make the same number of mutations found in natural variations, but random mutations, then you get something shown in gray, suggesting that these mutations may have erosion in the past, but they were selected again. So in the present genome, we only have these highly flexible sequences. So there was a really strong selection pressure based on DNA mechanics, we think, to maintain these extreme sequences. The student also then found a cousin organism, and in the mitochondrial DNA, there is another sequence that is also highly flexible, but not the same as this. And then he ran another search in the database using BLAST, and he found really interesting matches to bird chromosomes. And these are what's known as microchromosomes that are very tiny. less than 10 megabase in size. And here, the baseline is around 0 for random DNA, but you see all these peaks that go to 2s and 2.5s very frequently, and for these two different bird species. So somehow in these tiny chromosomes, birds have highly flexible sequences. And if you look at the closest outgroup, alligator, then you don't see that. You see basically just center that's 0 and just normal variation. You don't see really extreme sequences there. Another interesting point that we found is that if you calculate the average cyclizability average over an entire chromosome, and you do this for all of the chromosomes in the bird genome, this is for house finch, and then you get a really nice, interesting anti-correlation as you make chromosomes smaller and smaller, becomes more and more flexible. And this is pretty large range, almost 100-fold range in chromosome size. And we don't see that in the mouse genome, although here this version is much, much smaller, more like factor 2 or 3 only. And in fact, this effect that smaller chromosomes have much more cycles is actually pretty general for birds. If you look at all of the birds, 60 different species, tiny chromosomes tend to have very highly flexible sequences. So I think that we don't know yet. We haven't done the analysis whether this is something that is selected for. But one hint can be found. These are some nice photos I got from Google just to see what these birds look like. Is actually there was a paper last year about meiotic recombination in zebrafinch and other birds, songbird species, and when sex cells develop, they go through meiotic cell division, and this requires a crossover between the homologous chromosomes, and the presence of the crossovers are important for correct chromosome segregation during meiosis. And in fact, in their paper, they show that number of crossovers per megabase pair actually increases quite dramatically as you go to smaller chromosomes. And so we think that what we found in terms of highly flexible sequences for tiny chromosomes probably has something to do with the pressure to do meiosis correctly for correct chromosome segregation when you make such tiny chromosomes. So there's something that I'm interested in studying in the future. So here I can summarize that our discovery of extreme DNA mechanics in two unrelated branches of life suggests that our in silico engineering method based on an accurate prediction can actually predict extreme natural biology that nature has already found ways to utilize. All right, so that was the first part. In the second part, I want to talk about the relation between the nucleosomes and these compartments. Compartments known as A and B, and roughly corresponding to euchromatin and heterochromatin. Euchromatin DNA is not so well condensed, so you can more easily express genes, and chromatin is more condensed, and it's more difficult to gain access to the sequence elements hidden inside the DNA. So questions are, do a single nucleosomes know where to go? And do they know if they belong to A or B compartment? And do they actually encode intrinsically information for A and B compartments and gene level organization in their biophysical properties? Nucleosome is an electrolytic monster. Despite all the lysines and arginines with plus charges in the histones, we still have 150 electron volts of negative charges. So by themselves, they will not attract each other. But we know from many decades of studies that if you have polycations, such as polyamines, you can actually condense DNA or nucleosomes. So polyamines, putrescine, spermidine, and spermine with 2 and 3 and 4 plus charges with amino groups. In fact, polyamines actually abundant in our bodies, a high concentration of brains and internal organs, also in bloodstream. And it can actually make some of them, about 40% in our own cells. You can acquire them from food, and some of the microbiomes in our gut may also produce up to 10% of the polyamines. And within a single cell, polyamines are highly abundant in the nucleus and mitochondria and some of the uh, some of the organelles. So this is a very well-known study. So if you have DNA in a cuvette, and then you measure absorbance, and then when you keep adding polyamine, spermidine, it will eventually cause the DNA to crash out to the bottom, and so you don't see anything in supernatant. And only when you go to much, much higher concentration, there's a resolubilization of the DNA, and you recover this. But we don't actually have to think about that because it's such high concentration. So many years ago, my former colleagues in Illinois, Alex Mentip's lab with Jae-Jung Yu, they performed all-atom simulations of DNA in polyamine solution and trying to understand the basis for DNA attraction mediated by polyamines. In fact, they were able to calculate the interaction between the two duplex DNA molecules and show that interaction is negative in energy, meaning that there is indeed an attractive interaction. But that's something that wasn't expected was the observation that AT-rich DNA had a stronger attractive interaction than GC-rich DNA. And we were able to confirm this using single molecule FRET experiments done by Ha-Jin Kim. And this was interesting to us because in heterochromatin, we have about 60% AT content. So we have AT-rich region in heterochromatin, and gene-rich euchromatin tends to also have now high GC content, about maybe 40% AT and 60% GC. So one can fantasize that perhaps what we see here, polyamine-induced attraction between DNA molecules stronger for high AT content, may actually provide a contribution to nuclear organization into hetero and euchromatin. But back then, we couldn't really test this because it was done for just a few different sequences here and also on naked DNA, but without any histones. So this is where Sang-Wook Park came in, was a former student. He developed condensed pipeline. He grew mammalian cells and isolated nuclei. And then he treated this with MNAs to digest the DNA, linker DNA. And then he also went through additional purifications to purify homogeneously distributed mononucleosomes, single nucleosomes from the nucleus. And then he added spermine, polyamine to the tube, and then spun it down. And some of the nucleosomes that are more condensable, I guess equivalent to maybe sociable human beings, then they will crash out because they like to hang out with others who are also more sociable. And what's remaining in the supernatant will be the less sociable nucleosomes, less condensable. And by sequencing the supernatant and comparing to the input, we can actually determine how condensable each nucleosome is, for every one of the 30 million nucleosomes in our cell. So it's really nice. You can just, for every one of 30 million nucleosomes, you can assign a single scalar quantity. So here's an example of showing condensability across about 20 kilobase per size window using a genome browser. And you can see that condensability is actually usually a slightly positive. Zero is basically it's just the average nucleosome. And then we see these two major dips that are centered at promoters and CPG with promoters. And DNA is one, which is equivalently ataxic to show chromatin accessibility, shows a much narrower peak in the same position. So there is a relation between the two, but there are actually differences. Ataxic is very sparse. You have no information about anywhere else, but we actually have physics properties of nucleosomes across the entire genome. And our property is actually much broader in width, but this is really large decrease. And this is also surprising because instead of measuring things inside a cell, we are measuring this as a biophysical property of isolated nucleosome. But they somehow have properties that are consistent with less condensed chromatin that you expect from gene promoter regions. Here, we're plotting condensability across the entire human chromosome 1 in blue and compared to gene expression measured using RNA-seq, and we see this nice anti-correlation. Intuitively, this makes sense. Where you have high gene expression, chromosome should be less condensed and vice versa. But still surprising because we are not measuring condensability from inside the cell. There's no transfactors bridging different chromosome regions. We are just measuring them as biophysical properties of isolated nucleosomes. So that was really surprising. And if you go to the final scale to look at condensability across a promoter of many, many different genes, we see that for the top 20% of most highly expressed genes shown in purple color, we see the largest reduction in condensability and vice versa. And this is also rank ordering all of the genes here. So somehow we have a low promoter condensability for highly expressed genes. And as if nucleosomes know if they are in highly expressed promoters or not. And we also show that if you compare condensability in human embryonic stem cell versus differentiated cell, we see that for the genes that are highly expressed in the differentiated cells, then they tend to have lower condensability for the differentiated cells and vice versa. And so that shows that condensability is a cell type dependent property, and it's not just a function of AT content, because AT content is not going to change between different cell types. We can also correlate condensability in blue against the AB compartment score, and this score is actually basically a score as to how likely is it that that region belongs to the A compartment. So high value means more likely A. And you can see that this score, AB compartment score, is anti-correlated with condensability. So you have higher condensability, and then you get lower AB compartment score is more likely B, and so on. So this is also very nice. And someone has a question? Okay, yeah. So single nucleosomes actually know which compartment they belong to, but this information is contained in the biophysical properties. So one idea that we have is that the condensability can be the biophysical driving force of compartment formation. So imagine having condensability differences between nucleosomes, and some of them with high condensability may come close to each other briefly, but because of mutually high condensability. They stay together a little longer than usual, than average, and giving enough time for the third nucleation to come in to add to the initial combination. And then you can nucleate a cluster that can grow larger and larger, eventually seeding for the B compartment in red, and then blue becomes A compartment by way of exclusion from the B compartment. So that's the idea we have. In fact, Bin Zhang's lab at MIT, they ran MD simulations, Bryan dynamic simulations, where the only input is actually our measured condensability, and no sequence, no histone modification, nothing else, or high-C data, just condensability. And then you ran basically simulations of this kind, and then found that the predicted genome organization map and high-C is actually quite similar to the measured value, and with correlation better than 80%, suggesting that much of the information needed to form A and B compartment is already all encoded in single nucleosomes, and we don't need bridging molecules such as HP1 alpha and so on to get to that coarse grain compartment formation. So that was also very interesting. We then asked what are the genetic and epigenomic features of nucleosome condensability. So you can divide our nucleosome into 7 groups and see various correlations. And for example, you know that H3K27 acetylation is correlated with high gene expression. And then that modification is strongly enriched for low condensability fraction. And H3K9 trimethylation is correlated with poor gene expression heterochromatin, and this mark is enriched actually in high condensability fraction. So generally, we see that DNA sequences and histone modification marks, protein post-translational modification marks, generally showing correlation. High condensability show inactive marks, and high AT content, and vice versa. But this was a little funny because there are a lot of compounding effects. So we reached out to Tom Muir and Princeton, who created a library of histone modifications formed of the same exact sequence, but all different histone modification, and each containing a different barcode. So you can do condense-seq, use a barcode to decode what modifications was found in which fraction. And one way of presenting the data for polyamine-based condensation is shown here. And the red means that you have reduction in condensability when you have that modification at that location. And for example, this is the acetylation in K12 in histone tail 4. You can see that except for one modification, all other modifications make the nucleosome less condensable, just to different degrees. It's as if that the modifications are made to make the chromatin more accessible, generally speaking. In addition, acetylation has a large effect in terms of reducing condensability, and the effect is the largest for the tails in histone 4. And so that's something quite interesting. So I want to end by showing you some interesting data that we obtained with Erica Pierce, immunology and metabolism scientist at Johns Hopkins University. So she had created a mouse with enzyme knockout. This is ODC, and this enzyme makes putrescine from a precursor ornithine, and this is ornithine decarboxylase, and this is actually an enzyme that's inhibited using an inhibitor in some cancer therapies and trials, and but she made knockout mice in T cells, so you can deplete polyamines. And so what we did was to perform condensate from T cells in the wild type and the knockout mice. We're going to be focusing on just blue and green. And then this condensability metric shown here shows genome scale fluctuations. And let me zoom in here. So the wild type T cells show this blue condensability metric going up and down. But in the knockout T cells, the green, we see a much larger range of fluctuations. When blue is high, green is higher. When blue is low, green is even lower, suggesting that when you don't have enough polyamines upon polyamine depletion to translate the biophysical properties, condensability of nucleosome into meaningful genome organization, T cells tend to accentuate condensability contrast by enlarging the contrast. In fact, we have data really nicely showing that most of the condensability contrast comes from histone modifications. I'm not showing you the data today. So likely T cells will modify condensability through histone modifications to compensate for a lack of polyamine mediating function. And that's why you get this hyperpolarization of the condensability. And so again, this is a normal situation, but if you don't have enough polyamines as a mediating force, then T cells make redder and bluer. And we know that actually in politics, when you have society becoming polarized, deep blue or deep red, we know that causes dysfunction. And in fact, in the paper by Erica Pierce, they show that in T cells, if you deplete polyamines, T cells actually get confused about lineage fidelity. They don't know what they are supposed to be doing in terms of genetic expression, and that also causes dysfunction. So the idea is that when you don't have enough polyamines, then they do things to make adjustments, but globally it may work, but then locally there may be some issues because you do overcompensation that can be more detrimental in some genes than in some other genes. Right, so I want to end by discussing this slide. So I showed you this genome browser view of condensability showing two large minima at two gene promoters. And this is the accessibility marks shown in the bottom. And I'm also showing you RNA-seq data and various histone marks. And actually none of these actually existing marks can actually singly explain condensability. So we think that this is an emergent property, but it's a property that probably nature cares about because whether nucleosomes want to be more condensed or not directly physically, that is something that can control gene expression. And so we think there's a lot of room for improving the technology so that we can now more readily measure condensability as something that in epigenome properties that can be measured genome scale, different tissues, and different stress conditions. So this second part was done by Sang-woo, a former student, and with actually a lot of help from many other talented scientists, including current members in the lab who had to do quite a bit of experiments during the revision phase of the paper. And then we have had collaborations with people I already mentioned, plus Ben Garcia's lab at Washington University. So I'll stop here. And I mentioned, I think most of the people I talked about DNA bendability and condensability as a biophysical backdrops for chromatin function. And clearly these are not the most important properties, but whatever chromatin function that cell have to work with, we'll have to actually take these properties into account because they are there. And sometimes, maybe in the A and B compartment, this can be the main driver. But other times, can be supplemental. And on top of this background, we can also add trans factors uh, to have a more fine scale control to avoid non-specific uh, uh, distortions. All right, thank you very much for your time, and I'm happy to take questions.
Okay, thank you so much. That was fantastic. Um, yes, feel free to type your questions into chat, or if you want, you can simply turn your mic on. There's a little Zoom hand feature. If you want to raise your hand, that's fine. Um, but Q&A is open, so feel free to step up. It always takes a minute. I'm seeing a lot of clapping hands.
I'm going to stop share so I can see.
Oh, sure. Yeah, good idea. Okay. Okay, no one's going to be brave. It happens sometimes. Okay, well, I'll just say we'll give everyone a second if you want to type in a question or raise your hand. This was awesome. Oh, here we've got a few now. There they go.
Yeah, so actually, I see some questions on the chat box. Maybe I'll go from the beginning.
Sure.
Are there any features specific to regions where there is a disconnect between the prediction of A/B compartments and the high-C data? So that's actually a good question. We saw the discontinuity actually at the centromere in the human chromosome 1, and that region is pretty large. And so we and also the high-C data we compared to were acquired many years earlier. So it was before telomere to telomere genome was available. So there was missing information in the centromere regions. That's why there's a gap there. And interestingly, I don't know why, but we don't see that in the mouse chromosome 1. We don't see any major gap there. The second question is, when you study compartment for native mononucleation, how long is the linker DNA? So that is a really good question. So we are using mRNAs to digest the DNA, and we wanted to avoid over-digestion. And so if you actually just sequence our nucleosomes and then plot the length distribution, the peak position is actually at 147 base pairs, more or less exactly the same DNA that is bound by the nucleosome. And that size and that length distribution is not dependent on condensation. Supernatant versus condensed fraction, you get the same length distribution, so it's not due to length differences. All right, so then did you find any connection between condensability and DNA dynamics? Yeah, so we tried to see if there's any connection between DNA bendability and condensability, and we couldn't. So these are, I think, different properties. Yeah. And the next question is that contrast between A and B compartment in the simulation much larger than that in the high C map. Um, that's a great point. So in terms of actual experimental data, it's not just A and B compartment. You also have topological associating domains that are induced by cohesins and CTCF and so on, and many other factors. In fact, people have shown in the past that if you deplete cohesin or CTCF, then you don't have these contamination signal from these tests. And so the A and B compartment signal becomes even stronger. In fact, some signatures that are hidden by the topological association domains become visible when you remove these contributions. So that's one possibility why the contrast is smaller in the experimental data. And student from Bachman lab and the Schleifer's lab, AB compartment, in addition to that, whether you notice an association between nucleosome and condensability and TED formation. We didn't, and that makes sense because TED formation is dependent on trans factors, quiescence, and cis, and CTCF. So I think that prediction accuracy will break down in terms of high C prediction once you go to the TED size organization. One small question, is there a correlation between condensability resulting from the intrinsic physical chemical interactions with nucleosome and that aided by other chromatin regulated proteins? That's an excellent question. And we actually also performed condensing using, for example, proteins such as HBN alpha that we know to be important for heterochromatin formation. They bind to K9, H3 K9 trimethylation to bridge them. In fact, when you perform that experiment using Tom Muir library, we found that nucleosomes containing K9 trimethylation actually show up as the most, basically increasing condensability upon using that condensing agent. So yes, I think there'll be some differences for specific modifications. But generally, for protein-based condensing agents, we generally, otherwise, the contrast we see is much smaller than using polyamines, likely because you are distributing number of charges over a much larger entity than simple polyamines. There's another question. How does DNA methylation at GC-rich regions affect DNA bendability and cell type specific gene promoter condensability? An excellent question. So we have found, we have published this, in terms, if you have GC, if you have actually a CPG methylation, that makes DNA more rigid, less bendable, and that's something that we already published. And also, if you methylate CPGs, at least for the naked DNA, it actually makes DNA more condensable. And the genome-wide condensing data from the native sources also suggests that CPG methylation correlates with higher condensability. And that's also attractive because generally in facultative heterochromatin, methylation of cytosine actually can change from euchromatin to heterochromatin. So that also gives you additional possibility of controlling chromatin functions through DNA methylation. Next question is, do you think DNA condensability will be influenced by external forces on the cell? If the cells are compressed, you expect the high condensability areas to respond more to these external forces. Actually, that's a really good question. They may be more direct effect or indirect effect depending on how quickly you measure condensability after applying external force, and something that we can do and it'll be interesting to measure. And final question, super surprising to see almost all marks decreasing condensation, especially for methylation, which are typically associated B compartment. Do you think this could be caused by the modifications affecting the mononucleation size in the condensing protocol? Also for loop-seq values over chromosomes in various organisms, is that measured via moving window? If so, how large a window? Yeah, for loop-seq, we are measuring this with 50 base pair window. We can actually, when you measured loop-seq, we use a 7 base pair shifting window. We had 50 base pairs shifting every 7 base pairs across this genome. But once you train this, you can actually predict bendability with single base pair resolution. And you can basically 50 base window bendability across the genome of any organism that can be studied. And yeah, we don't think that mononucleotide size in terms of DNA size is different for different modifications because we see a pretty homogeneous property. All right, so there's another question about the Brownian dynamic simulation. I wonder, cation was how much important and potassium and what are the expected simulations or corresponding experiment for K. So that actually, that details are not known to me. And so the one thing that I didn't go into was that for the simulation, the bead size was not a single nucleosome, it was a 5,000 base pair bead, so roughly corresponding to about 20 nucleosomes. So perhaps detailed choice of ion or their concentration may not be as important that scale simulation. And then would you consider relating condensability to chromosome dynamics, identifying imaging techniques? That's an excellent question. I think because we have condensability information for every nucleosome, for major scale biophysical modeling of the chromosome, and this type of differential interactions across different nucleosome pairs will potentially help the simulation make it more accurate. And then maybe if you do cryo-electron tomograms of chromatin fibers inside a cell, maybe more condensed regions versus less condensed regions, maybe there's also a relation there. And of course, in the tomograms, actually, you don't know what part of the genome you're looking at. And so that's a technical barrier that needs to be overcome at some point, maybe by some of you guys in the future. All right, so I think I went through the questions very quickly, and yeah, thank you.
Thank you so much. That was the reason there was a pause. Everyone was doing a lot of typing for their questions. All right, great timing. We're right at the hour. So thank you so much for doing this. Really appreciate it. Thanks for taking all the questions. And yeah, thank you so much. Have a great weekend.